Good afternoon. So today we are also continue with the rest of the fluorescence microscopy. So last week we discussed about what is a fluorescence microscopy, how important that, and mainly why do you want to use antibodies, and we discussed about what is a primary antibody and secondary antibody. Why do you need to tag them or conjugate the antibody? What is the purpose that, and how important that? So these are all very important for fluorescence microscopy, even though the antibodies and other things is immunology, but we use the antibodies for to detect some proteins and other components in this fluorescence technique. So our goal is to get clear images as well as fluorescence, that is to see the proteins. That is mainly we have to handle the samples in proper way. So the first step is already we know that fixation, dehydration, infiltration, embedding, sectioning, and also staining. That is for already we discussed in the bright field microscope, the light microscope. So similarly, the fixation is that we define, that is to freeze cell and tissue in a particular time frame, so that all the molecules in that cell or tissues remain in its original location during visualization. So that is most important thing, so that then only you will see the prime. Also the antibody, we discussed what is a primary antibody, secondary antibody, and also different controls, that is to validate your images, and the results is based on the controls is plays in important role. So the fluorescence microscopy, already I said that the whatever light microscopic components, ocular lenses or eyepiece, objective lenses, stage, focus, condenser lens, the light source, all of them is currently, like a sophisticated light microscope has a digital attached to the computer and software. So this kind of microscope along with filters and other fluorescence lamps and software, that components all attached to the light microscopy, we can call it as the fluorescence microscopy. Here you can see the eyepiece, and then this is the lamp, fluorescence lamp. This is just digital imaging, but every company has a different components, like inverted microscope and upright microscope. Based on that, the places of the objective lens and condenser lenses, other components is in different assemble. But this is a typical different components, like fluorescence lamps, filters, then collector lenses, then tungsten halogen lamp house, then microscope, circuit board, the protect, the shield UV when you observe, and then objective lens, stage, this is eyepiece or ocular lens. So these are all the basic components of the light microscope. Sorry, fluorescence microscope. So what are we going to do with the fluorescence microscope? How we can utilize this technique, and how we can observe not only the structure but also expression of other proteins and other enzymes and other things. So one is to label the proteins. So if you really wanted to study a particular protein of your interest, so usually we can do some know the physiological and biochemical activities of these proteins. So to investigate the conformational dynamics and molecular interaction of protein or protein-protein interactions, and to track their trafficking in order to better understand their biological functions. So if you really wanted to know about how this particular protein is important for that components, in the cells or in the organ or whole the system, how it is important, and also how the particular protein is interact with the other proteins. So what would happen if it is a less expression, or what would happen if it is a more expression? How that will affect the cellular events or the functions of that other proteins. So, and also when you see how the proteins are translocated, you can track things that also, how important these proteins and expression in different cell types, tissues. This will understand the basics of that particular proteins functions and the biological functions. Also, to get better insight into the receptor ligand binding protein structures and enzyme activity, single peptide can be also labeled. So if you really wanted to study, the proteins structure and also how the proteins, the enzyme activity, or a single, the peptides, if you label all this, you can, with the fluorophore or dye, you can, get all these informations. Also, to ensure reliable results, the fluorophore conjugation should not interfere with the antigen binding characteristics of the antibody. So when you do, the protein labeling and other things, we have to make sure that the conjugate, the dye or fluorophore, it should not interfere with the binding of the antibody to the antigen. So that is the most important thing. Even though when you wanted to study a protein or protein-protein interaction or a peptide, if you wanted to label all this, it should not interfere with this binding of antigen and antibody. That is another important thing we understand. So we can also, label the nucleic acids. So the fluorescence-based assays, which play an important role in biophysical studies of the structure, function, and dynamic of nucleic acids. So recent advances in labeling methods and imaging systems have allowed the direct in vivo observation of DNA and RNA and their interactions with the other cell components. So now you have not only just the fluorescence microscopy, but also there are new, advanced microscope that will make, direct observation. So you can see a live image about, how the DNA and RNA and their interactions with other cell components. So the live cell imaging of nucleic acids has opened a new path for better understanding chromatin organization and gene expression regulations. So these kind of, techniques that the fluorescence labeling plays important role in understanding the genetic abnormalities and how it, studies a particular gene expression. So this will give you a more knowledge in depth, of proteins or enzyme or any, genes, and also nucleic acids. So you can see we can label the protein, we can label the nucleic acids, we can label the peptides, and also the polysaccharides. So complex polysaccharides such as heparin are structural components of the extracellular matrix. So these polysaccharides are essential for cellular adhesions, migration, and growth. So based on this technique, will help the compounds are also known for their anticoagulant, anti-thrombotic, anti-inflammatory, antiviral, and anti-angiogenic properties. So we can able to understand more, knowledge about all these compounds. The fluorescence labeling help. So the fluorescence-based method makes it easier to identify new bioactive polysaccharides and to characterize their biological functions. So the labeling is, we are discussing about, how these labels help us to understand on different, polysaccharides, lipids, enzymes, proteins, nucleic acids, and peptides. So another thing is the cellular lipids, which plays a crucial role in the cell for energy storage, for the formation of cellular membranes, and for intracellular signaling process. So the lipophilic dye, Nile red, is widely used to stain intracellular lipid in order to analyze their location and organization. Also, the lipids, labeling help to understand the lipid dynamics. Specific labeling in living cells is also possible to understand the specific, studies on the lipid dynamics. So far we discussed about, the fluorophores or dye, and also we discussed about the antibodies. We can conjugate that with the fluorophore or dye. How we can able to conjugate? That's the most important thing, right? So you need to know how these, antibodies or lipids, proteins, how we are going to conjugate that. Commonly used fluorescent labeling methods used by chemical, enzymatic, or peptide protein tag and genetic labeling techniques. So one is chemical labeling techniques. So the fluorophores dock to the target molecules through chemical modification, that is a covalent or non-covalent binding. So this method have several advantages as they are robust, easy to perform, and very efficient with a wide range of fluorophores. So this chemical labeling techniques is suitable for in vitro studies rather than in vivo. The other technique is enzymatic labeling. The enzymatic reactions allow fast, highly efficient, and selective labeling both in vivo and in vitro, which can be used to target proteins or whole cells. Yet, due to the large size of the labels, the interferences with the function of the target molecules can occur. So this is enzymatic labeling is very important, and you can study both in vivo and in vitro. But only the drawback is because of the larger size due to the labeling, the interference with the functions of the target molecules can occur. That is one drawback, but with the efficient understanding the correct concentration of proteins or whatever you're labeling, that will overcome these issues. We can do the the peptide. You can label the peptide and the protein with the tag. So recently developed technique allows a specific and selective labeling of proteins through the incorporation of short fluorescent tags which do not disrupt the folding or the function of the molecule. So this technique is also easy to perform and can be used to investigate different sites of single proteins depending on the peptide tag specificity. So very important, as we know that the peptide is a small fragments we sequenced, and also then we synthesize, and we can use this peptide for different purposes. One is you can purify the protein, or you sometimes in the tag and the peptide is a specific region for antimicrobial, anti-tumor, antiviral activities. So the purpose is, if you use this peptide, when you tag this, how we can determine this? Once you add the protein into this, for example, in vitro studies. So if you really wanted to study about the protein, how it kills cancer cells or other cells, when you add this into the monolayer cells, how this peptide is penetrate into the cells and interact with the other molecules. So what you can do is, when you label these peptides, then you can go under the fluorescence microscopy. You will know exactly how this, labeling, the cell peptide that will, go inside and where it binds. Also, the genetic labeling. The genetic labeling can be achieved using protein domains and also small peptides or single amino acids which are marked with the fluorescence dates can be specifically attached to sites along chromosomes in vivo. So this study also you can able to detect the chromosomal abnormalities and also deletions or duplications. Also, multicolor labeling is another interesting technique that used for the live cell imaging and for flow cytometry application, is the ability to follow or detect multiple fluorescently tagged proteins at the same time. So that this means, if you really wanted to study more than one protein, you can able to do the multiple color labeling. For this purpose, specifically designed dyes with a very large stock shifts can be used, which allow the simultaneous monitoring of different biochemical process. So, for example, if you wanted to study a single protein, then you can use one, a tag. But at the same time, if you really wanted to study more than one protein, how these proteins are interact with other protein and how these cellular events happen. So if you really wanted to study this kind of things, you can do multicoloring. So multicoloring means you can simultaneously detect more than one protein. So that means if you wanted to study 3 proteins, so what you can do is you had to tag or you had to conjugate the antibodies with the different color. So that means DAPI or HS or any other dye with a different range of color like a blue, green, red. So if that is the case, you can label each protein with the different dye and you can process. Then simultaneously you can see all 3 proteins where it is expressed and how it is, whether it is going decrease or increase. Everything you can understand this. So the multi labeling system helps to understand better for not only a single proteins or many proteins. So the fluorochromes or fluorophores dyes, it's very important in fluorescence microscopy and also associated with other techniques. It is biologically active fluorescent compounds that binds directly to the cellular structures. The chemical dye, which is most needs to be coupled to a macromolecule to be studied in the microscopy. Also, we can study the fluorescent proteins, which can fuse genetically to a protein of our interest. So we know how we can conjugate these, with the different dyes or fluorophores. The reactivities are target molecules, which classes and corresponding fluorescent dye reactions moieties. So here, we have a reactivity molecular class and reactivity moiety, and the examples of different fluorophore or dye. So if you really wanted to study the antibodies, proteins, and peptides, you can activated esters. Also, what are all the dyes we can use for this kind of study is alexaflor, scopolamide ester. Also, thiols reactivity site. That is, you can use the alexaflor 594 malmidine. That also you can, conjugate with the antibodies, protein, and peptides. Also, if you really study, the low molecular weight molecules like drugs, steroids, hormones, reduced sugars, metabolic intermediates. So if you wanted to study this kind of, things when you conjugate with the, alexaflor and hydrazide is one of the, fluorophore. You can conjugate to this and to study. So this is just, give you like how, the conjugates, the fluorophores and other thing with the antibodies and other components. So now we understand the fluorescence microscopy, and we know the importance of antibodies, and we know the what is a fluorophore, how you can conjugate with the different proteins, the enzymes, lipids, polysaccharides, and other things. So give an example like how we can do that in the laboratory. We know we conjugate the proteins and other things, and you understand the basics, but how we can able to do that in laboratory. So very simple technique, for example, if you really wanted to study any, in vitro studies, example, we grow, the cells, either human cell line or animal cell line. We grow that on the cover slip. So that's one of the method, simple how we can use. That is, so first you grow the cells in the laboratory, and then you can transfer that directly onto the cover slip. Or even you can grow the monolayer on the cover slip directly. Then you can fix the cells. As we know how we can fix the cells, it is with pyroformaldehyde or methanol or acetone also, and then washed 3 times with the PBS. So then, once you are done with the fixation, then cell permeabilization is important. So to facilitate that, you can use 1% Triton X. That's a detergent, plus bovine serum albumin. That is for which helps to prevent the non-binding sites for 2 minutes, so that then you can wash again 3 times. So this is like a typical method. So next is you have to staining. That is another important thing. So you grow the cell line, and then you can fix it, and then you can Triton X and BSA, you can do this, and directly you can label the cells with a blocking solution to minimize non-specific staining. So then incubate cells with the monoclonal or polyclonal antibodies specific for the protein of your interest. Then you can add the secondary antibody directed against the primary antibody so that it conjugate in the fluorophore. So when you look at here, this is the target of your interest. You wanted to identify this. So what we can do is first you can incubate the cells with the primary antibody. When you look at here, we didn't conjugate the primary antibody. So now the binding of the antigen and primary antibody, the interaction between these two has to detect. Then only you know whether the antigen is expressed or not, or how long, whether it is decreased or increased. So what we can do is you can use a secondary antibody that is conjugate with the fluorophore, and then this antibody will go and bind to the primary antibody because it has both the same IgG class, so that it will go and bind to these two. So this is a typical protocol. Anything, just a quick observation, you can do this. It is, you can do this a basic technique. So in order to stain the specimens to be visualized on a fluorescent microscope, it needs to be mounted on a slide using an appropriate mounting medium. So it is important when you have cells and you wanted to observe, it has to be mounted onto the slide with the mounting medium that will protect the fluorescence tag. So that you can keep it for a long time. So the mounting medium usually it's PBS and glycerol mix, and is usually commercially available. So for example, like the primary antibody, you can conjugate that with the sheep anti-P3 polyclonal antibody. That is the secondary antibody that is conjugated with the red Texas red. You can see red all the organs. So here also the primary antibody, mouse anti-tubulin monoclonal antibody they used, and FITC conjugated things you can see here green. So what fluorophore you use, depends on that. You can see the fluorescence color. So here you use red, Texas red, whereas here you can you use FITC, you can see a different green color. Similarly, the another method also used to detect fluorescence proteins. Here also the streptavidin is a bacterial protein that specifically binds biotin. This interaction may be used to label cellular components. So here also you can see guinea pig with the anti-insulin polyclonal antibody they use. Here also they use a rabbit anti-factor H. And here the mouse anti-glucogen they use, and this is a multicolor. You can see all these three together. So this is a typical multicolor labeling. That means you can see more than 2 or 3 proteins. So here when you look at this is the target of your interest. Then again it is same primary antibody and also the secondary antibody that is conjugate with the biotin. So the biotin and the streptavidin fluorophore, that is a conjugate the streptavidin that will be benefited. So here like If you really wanted to study this, the cellular events, this way, it helps more when compared to the typical, fluorophores. We will study details about the next class, that how important this each of them, how these antibodies important, and why do we want to use different techniques. And sometimes you can use some of them, the regular fluorescence microscopy, you no need to go for biotin and streptavidin, but this kind of technique will help you more when you go for other fluorescence microscopic techniques, that is flow cytometry and other live cell imaging and other techniques. We will discuss that more. What are the molecular fluorescent probings? That is most important, things you understand that. One is, dye. Some of the dyes is more important and specific. Which is directly binds, nucleic acids, which covalently bind fluorescent dye. You can use to purify the proteins. Also antibodies, that is immunofluorescent labeling with the primary and fluorescent secondary antibodies. Also you express in cells green fluorescent protein fused to protein of your interest. So this one, you can, the fluorescent protein, green fluorescent protein, you can tag them if you really wanted to study, and then you can clone it and express it and purify, and then you can use that protein for further study how this expression and things. And also the tag the protein. If you tag the protein, for example his tag, you can easily purify the protein using the his tag technique, and then after you purify, you can remove the his tag, and now you have a purified protein that is used for your other studies. So also you have a different fluorophores dyes. So here you can see it is all depends on the excitement and the filter, and the, you can see different colors. So here, Texas red, rhodamine, TRITZ, fluorescein, fluorex, true red, and hydroxycoumarin. All these probes are important, and depends on which one you choose, you will get this kind of fluorescence. Okay, again here also the one dyes, there are several Alexa fluor dyes. It's all depends on which one you choose, that will be see different colors. Also, if you really wanted to study the nucleic acid probes, and you can see different things that is directly you can target DAPI, HOICHEST, cytoblue, ethidium bromide, propydium iodide. So these are all X. So these are all directly if you wanted to study, you can probe nucleic acid with these fluorophores. Also different proteins. If you really wanted to study, we have several fluorescent proteins, that is, you can tag GFP, the green fluorescent proteins, turbo GFP, tag CFP. There are several proteins, that is, if you use these kind of things, you can you will get this kind of color. So our goal is, you wanted to study about protein, nucleic acid, and other things, how it is expressed and where it is expressed, and whether it is expression is decrease or increase. This will all, you will get it from fluorescence microscopy. Also, you can see the cellular events. Also you can see multicolor imaging, more than one protein or one other lipids, or you can able to see that as well. So this is more important. And also different probes and molecular weight and what are all them are important. So for exam point of view, I don't think you need to remember or anything with all this, but later I'm going to discuss with some of the important dyes. You need to know what is that for using. So some of the dyes are, this is the DAPI, that is a diamino amidino to phenylnidol. So that is, we simply call it as a DAPI, is a fluorescent stain that binds strongly to ATG rich region in DNA. Okay. Another also, the DAPI can pass through an intact cell membrane, therefore it can be used to stain both live and fixed cells. That is the advantage of the DAPI. Also, the HS dye is similar to DAPI in that they are also same blue fluorescent DNA stains, which are also compatible with both the live and fixed cell applications. Then EBFP, that is when the gene encoding for EBFP, which inserted into the host genome. That is another important things which the encoded proteins can enable both the tracking of tagged proteins and their expression level. So when you tag your protein with this, you can able to tracking of that particular protein of your interest and how that is were expressed. Also, hydroxy 4-methyl coumarin, that is a labeling reagent often used for preparing protein and nucleic acids conjugates of blue fluorescence. 4-methyl umbelliferon, that is the substrate recognized by beta-galactosidase as a means of measuring reported gene activity. There is another one, is a fluorescein, is often used for the in situ labeling of peptides, proteins, and nucleotide. Then the green fluorescent protein, this is also when the gene encoding for green GFP, which is inserted into the host genome. The encoded protein labels both the tracking tagged proteins and their expressions level, like EBFP here. There are different dyes, TRITC, rhodamine green, or red Texas red. These are all commonly used for many purposes. Aloxaflor, which also you can conjugate that to antibodies and proteins. The propodium iodide and cyto9. This also mainly used for live dead bacterial viability kits. So here also, when we can discuss this later, how important this. Another thing is the ethidium bromide, that is strongly binds to the DNA. So as you are all aware, the DNA gel electrophoresis, when you run the DNA after the, purify, to detect this, you can add the ethidium bromide and go and see under the UV light, you can see fluorescent band. So it strongly binds to nucleic acid so that you can see this under the UV light. So the fluorescence microscopy, very important to understand further that identification of cells and the cellular components and the monitoring of cell physiology with a high specificity. So like a flow cytometry. The flow cytometry is widely used in basic, research in clinical practice to measure the signal from specific fluorophores. Also, the cells and particles are analyzed and eventually sorted in real time as they pass through the light beam of detectors, which quantify the fluorescence produced by labeling antibodies or ligands. So mainly this technique is a broad application for many fields like immunology, hematology, transplantation medicine, oncology, and genetics. Another important technique is fluorescence in situ hybridization technique. That is, the fluorescence in situ hybridization, which allows the localization of specific DNA sequences on chromosomes. There is another microscopy called fluorescence correlation spectroscopy. That is, correlation spectroscopy allows the analysis of temporal changes in the fluorescence intensity of fluorochromes. So this technique is a sensitive tool for determining concentration of aggregation level of proteins as well as observing molecular interactions. So now you have, understand the basics of fluorescence microscopy. And also just we showed that, if you have a cells in the lab, when you grow, immediately you can go and see under the microscope for brief, technique. But for, the sections, like a typical, you can have a, to study, how that cellular, proteins or other components expressed in the cells, you have to process fixation, dehydration, infiltration, embedding, sectioning, right? So that during the staining process, now you have to choose whether it is go for regular or fluorescence. If it is a fluorescence, then you have to follow like this technique. So imagine that your sections, after you take a sections using the microtome, you transfer that onto the slide. And then you have to incubate the cell, with primary antibody. So that is your target here, proteins. Then you wanted to detect this primary antibody, you have a secondary antibody that is conjugate with the fluorophore. Another thing also you can see, primary antibody, the target protein is here. Again, the secondary antibody that is conjugate with the fluorophore. So the thing here is use antibodies raised against your protein of your interest. That is most important thing. And also it should be same species. The primary antibody is raised, against rabbit. It should be the secondary antibody also same species. Then only it can recognize. Similarly, here you can see if you use the primary antibody that is a produced mouse, then use the anti-mouse secondary antibody. So the simultaneously image multiple fluorophores to localize multiple proteins in the same cell is already we discussed. That is more than one protein of your interest wanted study, you can use multi-labeling. So the fluorophores for microscopy, fluorescein and rhodamine derivatives. So usually you can see the spectrum of light, from wavelength UV to IR. So if you use more than one, then you have to have a different, wavelength. That is, then only you can see a different colors. One is a fluorescein, that is nanometer is a 520, so that you can see a green here. Also, the Texas red 601 nanometer is, you can see a red. Whereas TRITC 53, this is also close to this red. So similarly, you have to choose a different fluorophores with a different wide range of colors. So then only you can distinguish. If you use the Texas red and the tetraxylene 2 TRTC, then you can see both are in red. You cannot say which protein is which. So that's why we have to choose the the different range with each other. So the fluorescence labeling is very, very important. So one of the technique is a FISH technique, that is a fluorescence in situ hybridization. This is the power of in situ hybridization can be greatly extended by the simultaneous use of multiple fluorescent color. So this technique that hybridizes a DNA nucleic acid probe to a target DNA sequence contained with a cell within the cell nucleus. Also, the variety of specimen types can be analyzed using this technique. So this is the method that is, we can commonly used the intact cells that are attached to a microscope slide using a standard cytogenetic, methods. So this is, the FISH technique. Your goal is whether you can choose one target or dual target or more than two. That is a multicolor. You can use it. This is a two color and this is one color, which allows one to look at multiple genomic changes within a single cell without the destruction of the cellular morphology. That is the interesting point for this FISH technique. So how we can prepare for our study is basic steps of fluorescent in situ hybridization technique is your targeted DNA you wanted to study, you have to have a probe, right? So we wanted to make a probe. So we, synthesize the fragments that is, integrating into your targeted DNA with the probes here. So what we can do is that the probe you can synthesize, and then you take the targeted DNA and then you have to make it with the denaturation. So once you denature this target and the probe, it will be, detached. And then you can make it this, it will go and bind to this target DNA here. For example, you can look at it here, the hybridization technique. And then once you are established them, make it the hybridization, then you can detect the probes. So you can see the target proteins, the probe, it is integrated into the target DNA of your interest. And then you can go this with the study in the sections, how it is going to be in the events. So this is a pivotal cytogenetic technique that has provided significant advances in both the research and the diagnosis of hematological malignancies and solid tumors. One is a diagnosis purpose. The FISH can be applied to detect genetic abnormalities such as characteristic gene fusion, aneuploidy, loss of a chromosomal region or a whole chromosome, or to monitor the progression of an aberration, serving as a technique that can help in both the diagnosis of a genetic disease or suggesting prognostic outcomes. So we can use this technique in two. One is a diagnostic purpose, the other one is a research application. So the diagnosis purpose, if there is any abnormalities in the chromosomes and other genetical abnormalities, you can able to detect using this technique. For research purposes, mapping or identification of novel oncogenes or genetic operations that contributes towards various cancer types. So the FISH technique is based on DNA probes annealing to specific target sequence of sample DNA. That is the you can target particular DNA, you can study that. In medical field, this technique gives more values and knowledge on different diseases, which includes histiocytide sweet syndrome, mosaicism, autologous fat grafting, differential diagnosis, mosaicism, streptococcus pneumonia, DDLPS, aneuploiditis. Also, the application of FISH in oncology, CML, MM, and pulmonary adenocarcinomas, prostate cancer, breast carcinomas, many cancer types. There are so many publications, so many values we get it from this technique for this. Also, there are a lot of different techniques we use with the basic FISH technique. So all related to RNA FISH technique, and also reverse FISH technique, importantly immunoFISH technique, that is association transcriptionally silent genes with other genes. Then DFISH, that is a two-color BCR-ABL probe that greatly reduce the false positive and false negative rates for fluorescence and in situ hybridization. Mainly it's a CML. So there are so many techniques, helps to detect, understand further with all these cancer types. So this is, Nikon microscopy data use different techniques, and how the fluorescence helps to understand not only just target protein but also organ or cells whole which express. Some genes or proteins are ubiquitously expressed. If it is you can target that protein, then the whole organs or cells will be expressed. So this is so epifluorescence illumination for stereo microscopy is GFP illuminators, especially larvae, nematodes, zebrafish, oocytes, and mature insects can be easily selected and manipulated when they are labeled with the GFPs. Also, live cell imaging techniques that to provide critical insight into the fundamental nature of cellular and tissue functions. Also, live cell imaging, that is, basic, techniques, analytical tool in most cell biology laboratories, neurobiology, developmental biology, pharmacology, and other related biochemical research disciplines. This, plays important role to understand basics. So pros and cons of fluorescence microscopy. So far we discussed about many advantages, and the techniques helps to understand basics, and also understanding the chromosomal abnormalities, genetic abnormalities, and you can detect the cellular events, and you can use to see the expression of proteins, polysaccharides, lipids, and everything, apart from the cell structures. The advantage is, this one is you can not only one protein or one color, you can do multiple. Also, you can do the FISH technique, and also this is not, very expensive technique. This is, compared to other microscopy techniques is expensive, whereas a fluorescence technique is compared to less expensive. Because of, you can use multiple color imaging, that means a different fluorophores you use that will interfere with your imaging. So also, if you have a thick sections, it won't be, um, properly expressed, because that will, reduce the quality of your structure imaging. So that is sometimes, this is uh, drawback for fluorescence. But nowadays, we have more technique experience, different, sensitive microscopes helps to solve all this. Also, like the multiple color, if you use the fish technique, also broad peaks cross over into one another. That is another important you need to know, because the range of color, that is you have to choose if you want to study more than 2 or 3. Also, because of the fluorophore, it will cover the expressed area, so that, the out of focus is possible, and also which reduce the, resolution. Also, the bleaching, the excited fluorophores react to become non-fluorescent if you keep it for longer time, because it's bleaching. Also, due to the phototoxicity, if you light source to observe this, that will also harm to the cells. Sometimes, autofluorescence or background, the cells have fluorophores too, that may look like once you want to examine. So that is another drawback, So that's why we wanted to use it controls. So if you have a different controls, then you can overcome this background and autofluorescence. So if you tag the protein when you clone, so this is protein called AAE protein, which is expressed in aggregated bacteria, actinomycetum comitans bacteria. It's outer membrane proteins. So we wanted to study about the protein of the AAE. So what we did is we cloned this protein, gene in, bacterial expression system. So we tagged the gene and DNA with the tag protein, and then, we clone it, and then we express it, and then we purify. So the purified protein, which helps the tag to purify this protein, and then we run onto the electrophoresis, gel electrophoresis, that is a polyacrylamide gel electrophoresis, run onto the gel, and then did the Western blot analysis, and you can see, the tag antibody we use to detect the protein expression. You can see that protein is expressed here. You can see that. So this way, when you tag the protein, you can able to purify, characterize, and you can also study the functions of these proteins. Also, like simple experimental, model. Usually, I will ask the students to do this. It's a direct, immunofluorescence sustaining of human buccal epithelial cells using monoclonal anti-pan cytokeratin, which conjugate with the FITZ antibody. So how we did this experiment, how the students always, we allowed to collect the buccal epithelial cells by scraping the cheek with the tongue depressor. So then, you can centrifuge and collect the buccal epithelial cells, and mix with the PBS, and 1 or 2 times wash it thoroughly. And then now we have a buccal epithelial cells in PBS. And then you take a, drop of, the buccal epithelial cells, and drop onto the slide. Then you can heat fix. So this is, very quick. So once you heat fix, and then you can incubate with bovine serum albumin for 15 minutes to block. So non-specific proteins were, binding on to the, this. When you use this, it will avoid. So then once you, block the, the buccal epithelial cells, and then you can wash again with the PBS 3 times. So now we have a monoclonal antibody that is anti-pan cytokeratin already, that is conjugated with the FITZ. Okay, so we know that the antibody concentration of this works. So we use this concentration and incubate the slides with the buccal epithelial cells already heat fixed. Then for conjugate for minimum 2 hours, and then again after that you can wash it. And now you're ready to go for observation under fluorescence microscope after air dry. So when you go and see under the fluorescence microscopy, then where you can see the buccal epithelial cells of fluorescent, because the anti-keratin protein is ubiquitously expressed in cells, so that you can visualize. So that is a basic, to understand what is the, method we can use. So this is, you are not taking cross section, or you are not taking any, different steps, sectioning, or this is a direct, just you wanted to see the buccal epithelial cells. So don't confuse with that, like a typical process is, if you really wanted to, do the cross sections, and that is a different technique. So here, the same similar method we did, we collect the buccal epithelial cells instead of anti-cytokeratin. We use AAE antibody. So when you look at this, this is a bright field microscope. The AA cells, it's like a donut. And whereas here, because of the AAE protein is expressed on the outer membrane surface, you can see the outer membrane is expressed. So this is a higher magnification. So now this study shows that the proteins are the outer membrane protein AAE is expressed in the outer surface of the membrane. Similarly, if you want to study buccal epithelial cells, you can see the whole buccal epithelial is a fluorescence because the proteins are expressed ubiquitously in the buccal epithelial cells, so that you can see the whole buccal cells become fluorescence. So this is, before we take this is a bright field microscope of this buccal epithelial cells. So this is a the basic understanding of how important the fluorescence, and how this antibody that conjugate with the how that will interact with the antigen, and then you can see their expression. So another important study is how this fluorescence microscopy can understand further to see live and dead cells of bacteria. So this is our study that in the laboratory we grow AA biofilm on the 6-well plate, and then we incubate diallyl sulfide, that is a compound, antimicrobial agent compound, and for 2 hours, and then we can wash thoroughly, and we let it go, this bacteria, for 2 days. So then it forms a biofilm, and then we wash it, and we add the live dead cell imaging kit that contains propydium iodide and cyto. So when you look at here, the top panel is healthy cells. We did not add anything. Just we grow the bacterial biofilm on the 6-well plate. Whereas the bottom one we grow, but we treated this with the antimicrobial agents, diallyl sulfides. Then we can use two different color panel here. One is a cyto, the other one is propydium iodide. So if it is a dead cells, that shows it's a more bright red color. Whereas if it is green or blue, green means that is all the cells are live. When you look at this, the panel, you can see the green colors, most of the biofilms are in live. But whereas here, when you look at here, the cells are treated with antimicrobial agents, all dead. That means it shows red color. Whereas here, very less, you can see, when compared to the control. Whereas here, this is the combination of these D and E. This is like you can merge using the software. You can see the dead cells is more, but live cells is less. But when you merge, you can see very interestingly, see the this is the biofilm, and the inside of the biofilm is less red when compared to the peripheral. You can see the more dead cells on the biofilm surface than the inside. So this we cannot see in the, without the fluorescence technique. So the purpose of this, the concentration of the antimicrobial agent is not enough to kill the biofilm. And also, which also reflect that it is also not penetrate deep into the biofilm. So this study suggests that one is the concentration is low, the other one is this antimicrobial agent is not enough to penetrate deep into the biofilm and kill the bacteria. So this is, you can compare this in the real world. When we have a, like a periodontal disease, the main reason is the plaque. If you are not properly remove the plaque, that will means you get more bacteria and it grow and secrete acids, or it, inflammation, then you cause, infections and, bleeding and other things. So if you not properly treat, with the appropriate concentration or the dose, this is what it happened. So, that's why the antimicrobial rinse, if you are rinse with antimicrobial rinse, if the concentration is not enough, it won't kill the entire biofilm, that is, millions of bacteria. So the fluorescence microscopy, information tell us how this response, each antimicrobials or antibodies response against the biofilm. Also, we wanted to study different things. So here, when you look at this, Parphyromonas gingivalis, it is another bacteria periodontal pathogen. So we wanted to study how this lactoferrin peptide, that is prevent the growth of, Parphyromonas gingivalis. Here also you can look, this is a control panel, and this is, the peptide with low concentration and the peptide with a higher concentration. So you can see in control, you have a lot of bacteria, whereas here, the number of bacteria is reduced, and also you can see a lot of dead bacteria. So whereas here the a higher concentration of peptide that kills most of the bacteria. So this is another way you can see visually, you can see how the bacteria are killed by this. Here also another dual species, that is Streptococcus mutans and Candida albicans together. How this interact. So this is you can see green panel, that is how this bacteria and fungus grow in the biofilm, and this is a red panel. So this is you can see the both Streptococcus mutans and the Candida albicans, and together you can see. So this is we treated with lactoferrin. This. So here you can see a lot of you can see the hyphae, that is Candida albicans, that will develop this hyphae, and a lot of the Streptococcus mutans kills, but you can see many spores, and also it prevent the number of hyphae as well. So this also reflects that the dual species, you can prevent the growth of Streptococcus mutans as well as the Candida albicans. So with this, we can complete this today. And next week, we wanted to show that 2 or 3 slides, important on how this Streptococcus mutans, Candida albicans, study, and also different fluorophores functions that also we can able to study, and how this like a fluorescence microscope is a help to understand different diseases already we discussed, and the insight to hybridization and more on. Okay, so we'll see you on next class and discuss more with this, and also we can start with the electron microscope. Okay, yeah, see you all next class.